Thank you. So our speaker tonight for the Phage Genomics Lab is Harriet Lee, and she will be talking about characterizing and identifying mycobacteriophages.
Hi, I'm Harriet, and I'll be sharing my screen. So hi, I'm Harriet Lee, and I'll be discussing what MCDB 1161, the Phage Genomics Lab, has been doing this entire semester. So overall, we've been working on characterizing and classifying mycobacteriophages. So mycobacteriophages are viruses that infect bacteria. And in this class, we sought to study and examine mycobacteriophages that infect Mycobacterium. Many students found phages harbored by Mycobacterium smegmatis from soil found here in Boulder, Colorado. There were also students who found phages at Kittredge Pond on campus. Here are a few of the final electron micrographs of some of the phages that students isolated. So there are two types of phages, lytic phages and temperate phages. Lytic phages undergo the lytic life cycle, as the name suggests, and temperate phages undergo the lysogenic and the lytic life cycle. The lytic cycle involves replicating phage DNA, synthesizing protein, and assembling mature phage parts within the bacterial cell. After this occurs, the bacteria is lysed and bursts open for new phages to exit and reproduce. Then the cycle will repeat itself. When undergoing the lysogenic life cycle, temperate phage incorporate their own genome into the bacterial cell's DNA and then remain dormant. However, when the bacteria is under stressful deteriorating conditions, the phage will excise its own genome out and undergo the lytic life cycle. So there are also two different morphologies of phages that we focused on for this class, Myoviridae and Symphoviridae. Myoviridae phages have short contractile tails. Symphoviridae, on the other hand, have longer non-contractile tails that are flexible. They also have two different types of heads, either isometric or prolate. And almost 90% of known mycobacteriophages have been Symphoviridae, and a majority of the phages that have been isolated from this class have also been Symphoviridae. So we may have isolated and categorized phages for this class, but why is it important? What use do these phages have? Well, in our modern world, we have an issue regarding the overuse of antibiotics and bacterial resistance to them. You could see the graph on the slide illustrates the rising percent of isolates that have become resistant to certain antibiotics. And over the course of 10 years, the percent of bacteria that are resistant to specific antibiotics have doubled or even tripled. And so the widespread use of antibiotics led to an environment where antibiotic-resistant bacteria can grow and prosper. And phages, however, may become a viable solution to this issue. Phages have the ability to target incredibly specific types of bacteria because their tail fibers are involved with the recognition of specific receptors on the bacterial cell surface. Phages then can be used to identify and infect bacteria that cause infections and help treat patients who might no longer have antibiotics as an option. Phages can also adapt and co-evolve with bacteria. So there's hope to send new phages that we've isolated in this class to databases, which then can help the medical field with infectious diseases in the future. So we first began with plating and purifying the phages. The plaques formed on a lawn of bacteria help us identify whether the phage was lytic or temperate. The image on the very left displays plaques that are clear, which means that the phages are lytic because the bacteria are immediately lysed and phages continue to infect and reproduce. The image in the middle shows plaques that have a clear center but a cloudy surrounding halo, which means the phages are temperate because some bacteria have been lysed while some still have their phage genome integrated into their DNA, some of the bacteria. And the image on the very right is an example of a quality control gel, which illustrates a way to visualize the quality and quantity of viral DNA. You can see how the bands in both sample 1 and sample 2 are very bright, which indicates that there's a lot of DNA. And the bands are also quite distinct, which means none of the DNA has really been degraded. So all of the phages, including the ones we've isolated, belong to a specific cluster. Clusters are a way to organize phages by nucleotide sequence similarity. Phages in the same cluster roughly have over 50% similarity in genomic sequences, and phages in the same subcluster have roughly 70 to 75% similarity. So in order to categorize our phages into clusters, we first had to cut the phage DNA at very specific sequences with enzymes. The DNA was cut with 6 different restriction enzymes, and the DNA fragments were separated by agarose gel electrophoresis. In the image here, we can see that the DNA was cut by both BAMH1 and HEY3 enzymes. To analyze and understand the cuts made by the enzymes on the DNA sequence, we had to use an external program called Phage Enzyme Tool. and we inserted the certain amount of cuts made by each enzyme, and then the tool gave us two possible clusters that the phage may belong to, K and B. Since digest patterns and bands are similar with phages of the same cluster, tools gave us clusters that also have similar amounts of cuts made by the same enzyme. So although we were given potential clusters by the phage enzyme tool, they still remain a hypothesis. We're not 100% sure if those phages belong to that specific cluster. So in order to confirm the cluster, we performed a polymerase chain reaction to amplify a specific sequence of DNA. There are specific primers that are designed to hybridize to DNA from a phage from a specific cluster or subcluster. The PCR here is visualized through gel electrophoresis. And so the phage enzyme tool most likely gave us information that the phage here belongs to the F cluster. So to confirm the tool, the F1 primer was chosen. The bright band that the arrow points to shows that the primer has hybridized with the DNA, so we can confirm that the phage belongs to cluster F1. And so there are many reasons why knowing the cluster that a phage belongs to is important. Phages in similar clusters may have similar host ranges, and their host ranges could possibly overlap, which means that it would be really helpful to know for phage therapy. And knowing the cluster can also help us better understand the evolutionary relationship among the diversity of phage and how they came to be. So there are many applications for phages that can be implemented in the future and are also being used currently, ranging from medical, agricultural, and even evolutionary applications. There are like several possibilities. So on the top right, you can see an image of someone who is treating a grapevine with a phage cocktail. Bacteria caused devastating disease in the grapes and several other plants. However, people were able to find phages that targeted those specific pathogens and were able to save the wine grapes. The girl on the left, her name is Isabel, and she was born with cystic fibrosis. This caused a sticky mucus to develop in her lungs, which harbored life-threatening infections. So in order to try and resolve these infections, Isabel and her family turned to phages. 3 phages were used, and they're right there to work against the infections, and they were also genetically modified in order to be more effective. One of the phages was actually isolated by a student from a university, which really emphasizes the importance of our research and our work. This therapy was injected into Isabel's bloodstream twice a day, and although she's not completely cured, phage therapy really helped Isabel regain a stable life, and now she's able to live life as any other normal teenager would. And there's a lot of hope for phages to be incorporated in many fields in the future to continue and save lives. So thank you so much for everybody who is part of this lab. Thank you to HHMI CPhage program professors Dr. Gild and Dr. Philman, Megan, Sabrina, Claire, Kylie, and all the other amazing TAs and PAs that helped the labs run smoothly, regardless of how this year was going and how it was a lot more difficult. And here are my references. Thank you for listening.
Does anyone have any questions for Harriet?